Happy January 4th. We're going to start today by reviewing your homework, reviewing replication, breaking the code. Starting with your complementary DNA strand for number 1, your code should have read A, T, G, G, C, C, T, A, C, G, G, T, C, T, A, G, T, 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 A, G. Hopefully you got that 100% correct. Following the complementary base pair rules, we're going to move on to molecule 2. Your strand, your complementary DNA strand should have read ATG, CCC, CCG, CAT, TGG, TGT, TGA. Moving on to strand number 3, the complementary DNA strand should read ATG, GAC, AAT, TCG, ATG, TTT, TAA. And the questions below, where in the cell does DNA replication occur, must be in the nucleus. Since the DNA never leaves the nucleus, replication will occur there. The steps of replication are very similar to what we wrote down in our notes, as well as what those videos summarized. The first step, you should have mentioned the origin, the location where we begin. This is where helicase begins to unzip the hydrogen bonds between the complementary bases. DNA polymerase will bind onto each strand, adding the complementary base pair rules following the DNA replication complementary base pair rules, A to T and G to C. Ligase, DNA ligase, is the important enzyme that will glue together both pieces, and at the result of DNA replication, there is a complete replica of 2 exact copies of DNA. Why DNA occurs and DNA replication occurs is because the new cell that is being created through cellular division needs to have the genetic instructions necessary for it to fulfill its function. DNA replication occurs so that that new cell has those genetic instructions. What is it called if an error is made during DNA replication? This is a mutation. Any nucleotide change within the sequence is referred to as a mutation. So, going through our 3 processes of replication, 3 processes of nucleic acids. DNA replication is the first. This is the full copying of all 46 chromosomes prior to cellular division. It occurs within the nucleus, and it will result in a fresh new copy of 46 chromosomes. Pause the video here and review video number 1. This is a little bit more complex than the other videos and adds in more enzymes than what we listed before. It's an accurate portrayal of the process and the structure of these molecules in the process. Please pause the video here and watch video number 1. Now that you've reviewed a video and the process of DNA replication, I think you guys are all masters at the enzymes involved in the process and the product of DNA replication. We're about to go into 2 other processes that nucleic acids are involved in that are exceptionally important in the function of cells. And these processes are referred to as transcription and translation. Transcription, so you hear the word script within the term, is when DNA is written into RNA. The term script literally means to write, and the process of transcription is when a single gene, a specific single gene along a chromosome, is copied into RNA. And this process occurs when that gene needs to, in some case, make a protein. So the process of transcription, once again, is a specific gene being coded into and transcribed into RNA. Following the process of transcription, the external environment of the cell is actually a very harsh environment. So the molecule of RNA needs to be protected, molecularly protected. And the process that does the protection is called post-transcriptional modification. So let's take this word and kind of dissect it here. Post we know means after. Transcription we now know means making RNA from DNA. And so this process is going to happen after a molecule of RNA is created. That RNA molecule is going to be protected from the harsh environment by receiving two things, a cap and a tail. And of course this has to happen inside of the nucleus because the DNA is there and it's a preparation for the molecule of RNA prior to leaving the nucleus. It does happen before protein synthesis. Translation is the last step we're going to discuss that nucleic acids are involved in. Now, when you think of the word translation, many people who are bilingual can translate one language into another, which is exactly what's happening inside of your cell. In the process of translation, the RNA that is now coming out of the nucleus is going to be producing and translated into a protein molecule. So when we talk about a protein, the organelle that should be coming to mind are ribosomes. This is the formal process of protein synthesis. This is when a RNA molecule is actually constructed into a protein. Now remember that the monomer of a protein is an amino acid, and the bond that unionizes those amino acids are peptide bonds. Since we are making a protein, this is going to happen within the ribosomes of the cell. So let's take a deeper look into the steps that nucleic acids and the processes nucleic acids are involved in. The first thing we need to discuss, and if you're following along within your note packet, I'm currently on page 8, talking about our RNA molecule involved in transcription and translation. So we know that the RNA molecule is classified as a nucleic acid. Its full name is ribonucleic acid, and it is made of monomers called nucleotides. All of this is a review from our nucleic acid section in Goal 2. The polymer, it is called, is a polynucleotide, and we're now going to learn the responsibility and structure of this molecule. The structure of RNA is actually dependent upon what RNA you're talking about. There's different shapes. There is transfer RNA, messenger RNA, and ribosomal RNA. So they abbreviate that as tRNA, mRNA, and rRNA. It is a single-stranded molecule. However, it does still have that phosphate backbone like your deoxyribonucleic acid. And the sugar within RNA is, as we know, ribose and not deoxyribose. There is a nucleotide, a nitrogenous base that is different within RNA. And the nitrogenous base of uracil will replace thymine in RNA. That means when building the complementary base pairs, we still follow the rule of G and C. However, anytime that we see an adenine, a uracil will be connected to that adenine. And the role of our RNA molecule is to move genetic information from the DNA within the nucleus to the site of protein synthesis in the cytoplasm, which we know the site of protein synthesis is the ribosome. So let's go a little bit deeper into each one of those three RNA molecules. Again, I'm on page 8, and we're going to start with M, messenger RNA. Our messenger RNA molecule its function is to read a gene of DNA and make a copy so it can actually leave the nucleus. This is going on the first line on page 8, directly underneath where it says messenger RNA and function. Messenger RNA's location, however, is within the nucleus, as well as it'll travel to the ribosomes within the cytoplasm. The process that messenger RNA is involved in is transcription, because it is the actual message. It's actually called messenger RNA because this molecule is what is transcribed in transcription, and it is the message that will be carried to the ribosome. It is also involved in the process of translation because the message will actually code for what specific protein will need to be made. So RNA number 2 is tRNA, or transfer RNA. The function of transfer RNA is a bilingual connector between mRNA and amino acids. The location that your tRNA molecule is in is the ribosome. This is where amino acids are going to be linked together with peptide bonds. And so since we're making proteins, the tRNA will be in the ribosome, in the cytoplasm. And the step that it's involved in is translation, specifically because transfer RNA allows for the messenger RNA to be transferred into the language of amino acids. That's actually why they refer to it as translation. If at any point in time in the video I'm speaking too quickly, please feel to pause and rewind and review. Moving on to RNA number 3. This is our ribosomal RNA. So just like you guessed it, this is actually something that builds up the ribosome and the ribosomal unit. Its function will be part of that ribosome where proteins are made. It will be located in the ribosomes. Now remember, those can be implanted within the rough ER, or they could be free-floating within the cytoplasm. And the process that it's going to play a role in is specifically translation. Since the production of proteins is the process of translation, the ribosomal RNA is very important in that process. So you can feel free to pause the video here and confirm that you have all of the notes down. So when looking at the structure of RNA, I'm going to show you mRNA first. So they show us here in this animation that this is the DNA molecule on the left and the RNA molecule is created to the right. You can automatically see that there's uracil in place of every thymine. Thymine does not exist in RNA. Uracil is the nucleotide nitrogenous base that is in your RNA molecules. And you can see that they're all made up of nucleotides, individual subunits to make the total polynucleotide. Our RNA, on the other hand, looks like a globular structure like this. It will make up part of the ribosomal unit, so we'll see that a little bit in more detail in a few animations down the road. And transfer RNA is quite honestly one of the most miraculous biomedical molecules you'll study. Evolutionarily speaking, this molecule has evolved to create this hairpin-like structure folded upon itself like so. Since this molecule naturally is within the cytoplasm, and like we mentioned, the cytoplasm can be a very harsh environment for organic molecules like RNA. This tRNA molecule has evolved to create this folding mechanism. And so this folding mechanism, or this pin hair, sometimes people call structure, allows for the nucleotides to be conserved and not damaged by the harsh environment of the cytoplasm. You also see that on the top of this tRNA molecule is an amino acid. Now remember, an amino acid is the monomer of a protein. And on the bottom of your tRNA molecule is an anticodon. So you've probably heard of the prefix anti, meaning against or opposite. And now we're introducing this term referred to as a codon. A codon is actually 3 sequences of nucleotides along the strand. And the code that we've been talking about, DNA, RNA, will be split up into 3s. Your tRNA molecule carries the anticodon. That's kind of a mnemonic device to help you remember. Moving on to their structures. Yes, complementary. Your anticodon is complementary to the codon on mRNA. So when looking at the 3 types of RNA, messenger RNA is really important in carrying the genetic information from the DNA in the nucleus to the cytoplasm. And again, it will carry the codon. The transfer RNA consists of a single chain of 80 RNA nucleotides folded into a hairpin shape, binds to specific amino acids, and there's 45 varieties of tRNA. Now remember, the anti codon will be located on the tRNA.